Okay, good morning, everybody. I'm trying stuff a little bit differently, and I'm trying to record this or post it as a live stream on YouTube. Hopefully everything works fine. We'll test it out, and if not, I'll switch it back to the way it was before. I had a little bit of an issue recording stuff. Okay, good morning, everybody. I'm trying stuff a little bit differently, and I'm trying to record this or post it as a live stream on YouTube. Hopefully everything works fine. We'll test it out, and if not, I'll switch it back to the way it was before. I had a little bit of an issue recording stuff. Okay, good morning, everybody. I'm trying stuff a little bit differently, and I'm trying to record this or post it as a live stream on YouTube. Hopefully everything works fine. We'll test it out, and okay. Maybe. Can you hear me? Kimberly, I can see you on my video screen. Thumbs up, you can hear me? Okay, okay. And I see Joe giving me a thumbs up. I don't know what was going on. I was getting I was getting feedback, audio playing over and over again. Yes, is the audio playing over and over again right now? No. Okay. I'm going to go back to share screen. And hopefully okay. So are you guys seeing the screen? You're seeing the slide with yes? I'm seeing Kimberly's head shake. Okay, good. Thank you. It looks like I believe everything is working right now. So hopefully, and this should, like I said, it should be going to YouTube Live. I think that was opened up at the same time, so it was playing both. I think I've got it under control now, maybe. So if this gets over and it does not post as it should, I will get stuff posted for you, previous lecture. All right, so starting off, I guess I should ask before I get going with the lecture, any questions or concerns before we start this? Get back to see if I'm not seeing anything in the chat or anything right now. So I'm going to progress with this. Let me go back to full screen. So chapter 5 deals with eukaryotes, macroscopic eukaryotes. There's all sorts of possibilities when we think about eukaryotes. And in this context, you know, typically we think about, in microbiology, we think about bacteria for the most part, and all of the possibilities of disease that they cause. And sometimes the eukaryotes get kind of swept under the rug. We don't always focus on this. However, there is some serious disease, some very deadly diseases like the one that we're starting with right here. So with that said, when we think about eukaryotes and their, you know, the microscopic eukaryotes and their potentials, there are all sorts of types of infections that they can cause, numerous possibilities that, you know, we want to kind of touch the surface on this morning. So initially, the first thing we want to think about is something that I assume most people know a little bit about. I also assume this is something that I would be willing to wager none of us have experienced. that's maybe not 100% true, but I think you hope you don't. So malaria is a very serious disease. It's something that has plagued humanity since, well, since antiquity. We have kind of literally co-evolved with the malarial parasite, and it has kind of shaped some of our evolution, and we it. It's pretty impressive. So malaria is a type of protozoal organism that's transmitted by mosquitoes. It's transmitted by the Anopheles mosquito, and it injects this sporozoite stage. It says that it's this and the center, but really the sporozoite stage is this one right here. And with the saliva from the mosquito, it injects these sporozoites as they get into our circulation. Well, there's multiple things that truly need to happen, and we're not going to go through the real life cycle of malaria this morning. But ultimately, after a brief visit to the liver cells, they replicate and are released into the red blood cells, which is what we're looking at right here, to in turn infect these red cells. And we can see a handful of different stages of red blood cell infection. We call this the erythrocytic cycle. And in this, we can see all sorts of variation. So initially, as the parasite infects, we get these ring forms that start to emerge as the parasite starts feeding on the contents of the cell. You can see it kind of a little more pronounced right here. And as they start degrading stuff, all of these little granules, the speckles inside of the cell, we call that hemozoin. It's degraded product of the cell. Of our own cells. And as that cell's in there replicating, it goes through multiple rounds of asexual replication so that a single cell generates many offspring out of that. And as this is occurring, we call these schizonts, where we've got these replications occurring. That would basically fill the cell up with baby malarial organisms. Plasmodium is the genus. It fills it up with a lot of these guys to the point that the red cell explodes, releasing them out to in turn infect more cells. And the cycle continues. You get series of fevers and chills as a result of the immune response and the recognition of those cells when they're on the outside. Once they get inside of the red blood cells, the response starts to subside a little bit. Typically, the course of infection in the red cell happens a couple of days. So there's definitely more details that we'll go through later as we start thinking about this specifically. So there's all sorts of possibilities that we think about trying to protect ourselves. If you are, if you happen to be in an area where malaria is endemic, beds with netting around them, mosquito netting, is one mechanism to help prevent the mosquitoes from coming in and feeding. DEET-based insect repellent, wearing clothing with long sleeves, pants, things like that, that hopefully protect your body from being fed upon by the mosquitoes. But that seems, maybe not 100% effective. And ultimately, this is a battle that we try to fight, but it seems as if we're almost in a losing battle with these guys. It's very difficult to think that we can truly be protected. There are definitely some drugs you can take if you're going into areas where malaria is endemic to hopefully prevent contracting it in the first place, but living in these areas definitely sets the stage for issues. So enough of that right now. This is something that I think is really pretty interesting. It's something that oftentimes, we hear about this and we get some ideas that are really wrong. So this image coming out of the text is actually the one of the text boxes, the clinical focus, talking about a person that, well, contracts this, and, you know, multiple things happening in here, wondering what's really going on. Just a second. So in the text, it says 7-year-old girl with this spot on her arm. We later will describe this as ringworm in the text box, which we're showing it right here. This is something that's pretty common. It's common with little kids, not that adults can't get it, but it's common with little kids. It's common with household pets. Definitely a common occurrence, common infection. It's something that has been, well, it was initially thought of as a worm because it kind of looks, as you're looking at this image, it almost looks like there's a worm, kind of a concentric worm right under the surface of the skin, but that's really not what's happening. We know now that ringworm is actually a type of fungal infection, a dermatophyte, so a fungal infection of the skin, but it has been called ringworm. It's been, its genus is Taenia, meaning worm, but again, it's not truly a worm. So the text goes through and talks about some of the details with this in the text box, kind of following it through. They use, as we look at this, one mechanism that can help kind of get to the bottom of it is a tool called a Woods lamp. It's really just like a black light, really. It's a type of UV light that you can shine on the skin, and if this is actually ringworm, it will fluoresce. So kind of this little circle glows with that black light, and that is pretty indicative of that. Put some creams on there, some topical antifungal agent on the lesion, and this is something that requires weeks of treatment to truly kill the fungus, and we'll talk about fungal infections and all of that later on. So malaria to ringworm, now to one of my favorites, Giardia. This is a type of flagellated protozoal organism that is in the United States. It is a very common protozoal infection, probably really among the top most common types of protozoal infections. It's something that maybe a good portion of the time might be described as asymptomatic. You could contract this and not even recognize it, but if you do have symptoms, you know, there's definitely some issues with this. We contract it by drinking contaminated water. Typically, that's what we think of. It could be ingestion of some food where the water has contaminated it, whatever. The Giardia, here we can see two images. Both of these images are the active feeding stage of the parasite, what we call a trophozoite. And trophozoites are a name that's used with all sorts of these. It just means the feeding stage. So if you say trophozoite, that doesn't mean we're talking Giardia. It just means this vegetative, the feeding stage, all right? So you can see here in the intestine, they are all over. And the potential for these guys, they replicate. And as they replicate, they basically just start taking up the surface area within the intestine, causing some pretty serious issues in the GI tract, absorptive issues, so you get malnourished. And you also have some pretty serious diarrhea as a result of this. So there are lots of kind of symptoms with this that really start showing up in a human almost a month after you've become infected, because it takes time for these little guys to replicate and to take up space, right? But once they have really done that, the potential for some serious infection is definitely present. They say with some of these guys, a single a single bout of diarrhea can allow you to release upwards of 14 billion of these parasites, which is just a mind-boggling amount of them. In order for the life cycle to be completed, it's not the trophozoites that need to be released out into the environment, it's the cyst stage. So they're kind of resting stage. That's what's infected to us. It's the ingestion of those cysts. So if these guys are able within the colon for moisture to be removed out of there so that, you know, that the stool is solid instead of liquid, they form cysts. They're released into the environment, and again, through contamination, you contract that, and there you go. This image in figure B shows the ventral side of the parasite, and right here is a huge sucker. This ventral sucker takes up almost the entire surface, undersurface of the parasite, and that's what attaches to the intestinal epithelial cells. This again, electron micrograph, so we can't see it, but they've got two nuclei, and seeing images of that where they're stained up, it almost looks like kind of a smiley face with their double nuclei in there. But I always jokingly say, if you get it, you're not smiling. You're pretty sure you're dying. That's what you think. Another type of parasite, and these guys have these sporozoites that are similar to sporozoites, thinking malaria, but this isn't malaria, all right? So we see oftentimes with these protists some similar stages in the cycles, and the sporozoites are juvenile reproductive stage, okay? So Eimeria is a parasite that can infect us, it infects other animals as well. With this, there's a couple of potentials, and here you can see these oocysts. So these are like the eggs. These oocysts are shed in the feces of an animal. As they are released out, initially they are what we call unsporulated. So they're not, really what we mean by that is they're not fully developed. And so these unsporulated oocysts, at this point, they're not infectious. And if you happen to ingest this stuff, at this point, you're not going to get infected. But over the course of time, a few days outside of the body, in the environment, the oocyst is able to sporulate, so it starts to develop, if you will. And we get to the point where we say it's a sporulated oocyst. At this point, it's infectious. So now we've got inside of this cyst these little portions of the Eimeria reproductive structures that we call sporocysts. Inside of the sporocysts, as you look at them right here, it almost looks like a little kernel of grain. There's two little bits. There's two sporozoites inside of each of the sporocysts. And inside of this oocyst, there are 4 sporocysts. Holy cow, there's a lot of stuff going on. So each of these guys has the potential to release out these sporozoites, and the cycle starts to continue. This is after we've ingested these guys. So you ingest the sporulated oocyst, it gets into the gut, that causes the shell of this to be broken down, releasing the oocysts to release, or breaking open the oocyst to release the sporocysts, which in turn basically hatch out, if you will, releasing those sporozoites. So there's multiple things that happen within here. And in turn, these cells get, just like we were looking at before, where in the malaria, where they get into the cell and they start developing. Again, we're not going to go through all sorts of steps with the life cycle. Some of this is kind of similar again, but we go through rounds of replication, producing these schizonts, replicating inside of the cell, rupturing it, releasing the next stage that we call merozoites. And that's also something that you see as a stage with malaria. These merozoites can be reinfective, going back to a new cell, a new red cell, and infecting it. And oftentimes we see these cycles kind of replicating a few times before we progress to this next step, to the generation of the sexual phase of these. This is all asexual replication. So we generate some gametocytes, so male and female sex cells, if you will. It takes a handful of replications asexually to get the population up sufficiently to kind of trigger the progression of this next step. So some of these cells, instead of forming merozoites, form the gametocytes, which in turn, this is the sexual phase, these gametocytes form, and they in turn come together, they unite, forming the zygote, and this syngamy is this sexual union, generating the oocyst, and basically the cycle is continued at that point. Lots of stuff, all right? So asexual, sexual, and then the development of the parasite in the environment. 3 kind of steps required to complete the life cycle. So there are lots of potentials when we think about the protists and the possibilities for infection. And sometimes when we think about them, we try to group them up or classify them based off of motility. And there are we're showing 3 ways that they can move. There are others that don't move at all, right? And that, I guess, maybe should have been figure 4 or figure D, but that's okay. There are ciliates with short little hair-like projections. This is a paramecium, something that's not pathogenic in us, but Balantidium, that we'll see here in a bit, is a ciliate that is parasitic in us. Here's an amoeba. This is just a general one, maybe amoeba proteus or something like that. Entamoeba would be one that is parasitic in us. So these guys move, these guys move by pseudopods, so projecting out their cytoplasm and pulling themselves kind of through their environment. They have an outer bit called ectoplasm and an inner bit that is more liquid-like called endoplasm. So endo and ectoplasm around there, and they push out the pseudopods, like I said, and move around. And then this last one, this is a flagellate, so flagella, and there are some potentials for multiple arrangements with them. Just kind of depends. So cilia, pseudopods, and flagella are the 3 mechanisms of locomotion with these protists. And there are, like I said, other protists that lack the ability to move. So we think of them maybe, well, just as their own separate, but there are a lot of variations with them as well. So this image is a phylogenetic tree that I think is interesting. It shows a lot of details here, but it's something that is, we're still speculating, is this accurate? Can we make it a little bit more refined? I think we're hoping that we can actually do that. And this is, as we've got it, we've kind of separated it a little bit more as we've sort of color coded. There are numerous protists that find themselves grouped up throughout. And with that, definitely the potential to kind of start thinking about maybe a little further separation. Historically, we've grouped the protists based off of some characteristics. We've said there's some that are animal-like, there are some that are fungal-like, and there are others that are plant-like. So really 3 different, very different types of groupings. But we need a little more. And as we see here, we've kind of further divided this as we move and think about really this complexity. So there are handfuls of them as we move through here. The dotted bits are some suspected lineages as we move through here. Again, a lot of the variation in the protist begs the question to needing to further separate this. So as we look through here, my pointer's down here at the bottom, so we'll just start right here with the Epistha Cantata. This is where we find ourselves, right? Right within here. We also see the fungi, which we'll hit a little of these guys at the mid part of this chapter. So a little bit here that we think about. Entamoebas, we will be thinking about in here. Little bit of detail about some of the algae and their potentials for issues, toxins that might be secreted, right? These guys, Rhizaria, we pretty much skip over. The top two, maybe a little bit more detail as we think about these guys, right? Diplomonads, these like the Giardia we just looked at, euglenozoans, there are some pretty serious trypanosomes that fit within this category. Apicomplexans, this is where our malaria falls out. The ciliates, I showed that ciliate a minute ago and said Balantidium. The top two are where we see a lot of them, and there's a couple of extras, all right? So a lot of variation, and they group out in definitely different supergroups, showing not a distinct evolutionary relationship with it, more branching, right? More variation, definitely more diversity with that. So these next couple of tables breaks it down into a little more detail, and it also provides, with some of them, provides some details as far as if we've got parasites or not, or other potentials. So the Excaviata, Fornicata, these guys, cyst formers, the double nuclei, no mitochondria. There's some interesting thoughts about some of this stuff as far as why these eukaryotic cells wouldn't have mitochondria. There's kind of two schools of thought. One thinking that maybe, they're parasitic. They are maybe bathed in nutrients. They don't need to fully metabolize, so they lost the mitochondria. Oftentimes in parasitism, we see reductions in some of these. And so maybe that's the case. There's other ideas that maybe in the process of this endosymbiosis that we described in the last unit of material, maybe these groups of organisms, maybe they diverged before that occurred. And so they never had mitochondria in the first place. I don't know. I think maybe that seems likely. So Giardia is our example. Giardiasis is the infection. The parabasilids, the trichomoniasis is an example with that. Trichomonas vaginalis is the organism that we'll hit a little bit later. These guys, we don't see cyst stages, so it's a direct transmission to the tropozoite stage is what's transmitted directly. The euglenozoans, as I described, the trypanosomes fit within that. So the euglena is photosynthetic. The rest of these guys are not. Heterotrophic and flagellated, even though maybe with the leishmania it's almost inapparent. At least at one stage, it definitely is. So trypanosomes cause some serious issues: sleeping sickness and Chagas disease. Leishmania causes leishmaniasis, all three of which really some pretty nasty diseases. Chromoalveata, we've got the dinoflagellates. These guys, we can see some red tights. This has some issues with toxins being secreted that can make you sick if you happen to eat some fish or shellfish in the, with one of these blooms, if you will. Alexandrium, this is paralytic shellfish poisoning. and Pfiesteria, this is another algal bloom issue. The apicomplexans, I mentioned malaria, so Plasmodium right here is one. These are intracellular parasites, so they get into cells, like into the red blood cell as the example. Cryptosporidium is a pretty nasty diarrheal disease, cryptosporidiosis. Thorellia or Babsia kind of together, and I think really we're shifting to Thorellia. Toxoplasma is another one that's an apicomplexan causing toxoplasmosis. That's something that if you're a cat owner, you maybe know a little bit about that. We'll hit that later. As far as the ciliates go, Balantidium is the only one, the only ciliate that we think of infecting us, causing balantidiasis. There's a couple of others. We showed Paramecium as we started, and Stentor is another one that I think I've got an image of that here in a minute. I'm pretty sure there's some videos of these guys that will really show some of the stuff where they're moving around and things like that that are pretty impressive. So I will see if maybe I can find some of that and post it. I do know there's a website, it's YouTube, Journey to the Microcosmos, that shows all sorts of videos with some of these guys. So if you're interested, you can search that out. I will try to find one and post it on there, or at least post the title of it so you can search it out. The last one on here, the oomycetes, these guys, water molds, it's more of an agricultural problem. Phytophthora is an example with that. And we can see issues, again, agriculturally. The rhizaria, as I was showing that figure before, I was like, well, we'll kind of skip over this. Again, it's, yeah, there's nothing that we think about issues except agriculturally with the Cercozoa, Spongospora, subterranea, excuse me, sorry. These guys, powdery scab with potatoes. We're in Idaho, so maybe we should mention that. So this next one is not something that we think about as issues, but maybe benefits. So Archaeoplastidia, the red algae, these guys have a handful of things in here that are potential benefits for us, and especially as we look in here, maybe the starch, but the agar as well within the cell walls. This is something that we utilize for our agar in the labs to solidify and keep things solid at like body temperature. So it is a huge benefit, and it's recognition of using it in microbiology really lends to, well, it was initially, and it still is used in cooking. So in making types of desserts, you can add some agar. It's like adding gelatin, but the agar makes it stay more firm at warmer temperatures, and done deal. That opened up all sorts of avenues to being able to cultivate things that we weren't able to deal with before. And we mentioned a little of this in the previous unit when we were talking about, you know, different types of media. But these are the two sources that we get it from. So there you go. The amoebozoa, these guys, there's some slime molts, dictyostelium, and I think one of the next figures shows the dictyostelium. It's not something that's, you know, an issue to us, but it is definitely pretty cool. The ones we want to think about as far as issues are the enthamoebas. There's both trophozoite and cyst stages with them. Entamoeba causes amoebiasis. Naegleria is one that we think of as, well, we'll go through details with these guys later, but it gets into the brain, causing meningoencephalitis, primary amoebic meningoencephalitis. We call that PAM for short. That's better. Um, oh man, some of the, yeah, I feel sorry that you have to spell all this stuff out. The acanthamoeba causes issues with eyes, so keratitis, and it also can affect the brain, causing granulomatous amoebic encephalitis. It's different than the Naegleria. So we'll hit both of these guys in a little bit of detail later on. Pretty crazy stuff. I'm assuming most all of you have heard uh, about the brain-eating amoeba. That brain-eating amoeba is this guy right here, the Naegleria, and it is something that is, to me, pretty terrifying. It's an organism that, if you happen to get it up your nose, uh, your prognosis is very poor. We have only saved barely a little over a handful. I think we maybe are at 6 people now that we have been able to save that have contracted Naegleria. Basically, if you get it, you're going to die. That is terrifying, and one that makes it one of my favorites. That's pretty stinking cool. All right. And lastly, the Epistha contata, the fungi. We'll hit some of these guys. They're a little different with the chitin in their cell walls. They can be unicellular or multicellular. And there's handfuls of different potentials. The Zygomycetes, Ascomycetes, so like our yeasts, the Candidiasis, Basidiomycetes, Cryptococcus fits within there, and the Microsporidia. So 4 different types of potentials and different types of body sites that are impacted. As far as moving down into the last bit, the animals, there are some that fit within here, multicellular, heterotrophic. We think of the helminths, the worms. So these guys can be huge, but we still think about them in microbiology because, there are stages that are definitely microscopic, and typically thinking the infectious stage, it's usually an egg or a cyst stage that has been ingested, and usually these things are very small, often microscopic. The adult stage of the worm, however, might be disturbingly large. There are others, like maybe the schistosomes, that are barely visible with the naked eye as the adult stage. So kind of depends. Tapeworms down here can be some of the big ones, having like a beef tapeworm of maybe 5 meters long. That's pretty impressive. Okay, so the slime mold that we mentioned, Dictyostelium discoideum. These are really pretty interesting, pretty cool looking, and here seeing one out in the environment, kind of impressive seeing this. So it's individual amoeboid cells in this first figure, and you can see it kind of branching out, pushing out those pseudopods and moving, pulling itself through the environment, right? They in turn can aggregate together, and we can also see as they start to aggregate, they are single cells, but they all start kind of coalescing and coming together to form a very primitive multicellular sort of individual, right? They are still single celled, but they kind of start acting as a unit, which is pretty impressive. So life cycle with these guys, let me see if I can move this a little bit so I can see it. So the life cycle is typically individual cells, right? Individual amoebas. But then as time progresses, they will come together, right? Giving this multinucleate, so many cells together. So it just depends. So we start off here, and we're looking at this where these fruiting bodies are forming. It's all kind of aggregated together. It can be 10 or 1. So we've got these fruiting bodies within here. Here's a cartoon image of it. So it shoots up these little stalks, and these fruiting bodies. It's almost like the fungi as they're producing the fruiting bodies, the mushrooms, uh, in their process of replication. So it produces these fruiting bodies. Inside of the fruiting body are the spores. These spores, again, reproductive structures. Through the process of meiosis, we generate these mature spores. Here we show the haploid cells, right? The spores are released, they germinate, and that gives rise to our amoeba, right? Which is haploid, right? So they uh, they grow, they divide, they form more individual cells, and then we've got the potential for two amoebas to fuse and form a zygote, right? Uh, so as this happens, we now, going down this way, as we fuse together, now we are diploid, right? And we go through more meiosis, multiple rounds of mitosis, and now we've got haploid amoebas that are released. So it's only a brief period of being diploid. More haploid cells, back to this, and basically living in the environment, cycle continuing. There are potentials after this where we see this, that they aggregate, forming kind of this slug. It looks almost like a bright yellow slug, and it's able to move through its environment at a slug pace of about 2 millimeters an hour. They then stop migrating and shoot up their fruiting bodies, and the cycle continues. Pretty interesting. This similar idea with the plasmodial slime molds. So here we've got large multinucleate over here, all of these little purple blobs within here, and then producing the stalks. Here we've got a longer period of time of being diploid. So you can see kind of an alteration of generations here with the diploid cycle and the haploid cycle. And it's different as far as what's happening. So there's also potentials up here with these diploid cells. If the habitat's not suitable, they go dormant, forming the sclerotia that allows them to basically maintain, persist. And then when the habitat gets back to being more suitable, they're able to go back to this more vegetative stage, if you will. So after that, I'm not going to go through all of the details. It's similar to what we've kind of talked about, but producing the sporangium, releasing the spores that are haploid. Here we can see amoeboid and some flagellated gametes. So these guys with a little bit of variation with this. These guys have the potential to convert back and forth between the amoeboid and flagellated forms. And then dependent upon the types of cells, they can unite, fusing, fertilizing, going up to the diploid stage, and the cycle continues, right? This is not the same as Naegleria, but Naegleria does exist in amoeboid and flagellated forms. Kind of similar idea like that, and it does have a potential to shift back and forth between the two, maybe lending to a little bit of evolutionary relatedness that way. So the apicomplexans, we saw this guy before as we started the chapter talking Plasmodium, the malarial parasite. Apicomplexans would have been the ones on that figure that I said, well, we need the fourth one. We need letter D up there for those that don't move. Apicomplexans do not move. So no flagella, no cilia. They don't move by amoeboid sorts of projections. They don't move. They get into your circulation and use it to allow them to travel around, but it's not they don't have any sort of means for locomotion. They have, and I feel like this image got cut off because I feel like this is probably the apical end where there's more detail on it. This does not seem to be the right end of it. All right. So the apical complex, this tip of the parasite, has a handful of reservoir pockets that hold all sorts of different types of substances that allow the parasite to penetrate into host tissues. They have other substances that act kind of like I think of it like a heat-seeking missile as an example. They allow them to sense, to smell the specific portions, properties in a liver cell that allow them to hone in on that and to penetrate into it and start the first bit of the infectious cycle. So pretty impressive. And so the apical complex has lots of substances that in turn lends to their success in establishing infection. And again, they do want to be inside of cells. For the most part, there are definitely times that they exist out. Okay, next, here is Balantidium coli, and you can see this image, the cilia all surrounding it, little hair-like projections, and it is, first off, it's huge as far as protozoal organisms go. It's one of the largest protozoal infections, size-wise, that we see, and it's the only ciliate that's capable of parasitizing us. So we can see other, I'm sure there are other ciliates that we might see maybe living in our gut as a commensal. So something maybe feeding on bacteria and things like that, but not causing any sort of illness. It's only these guys that cause disease. And something that's at least here not really very common. All right. So with that, I think this is a pretty good stopping point. Let me make sure. Yeah, let me talk about this real fast, just because it's more ciliate, and Balantidium was really where we were at. So here's, this is Paramecium, so non-parasitic, but similar idea as far as Balantidium, what it's doing. These guys have an oral groove, so like a primitive mouth. They have also an anal pore where they are able to release wastes. The cytoproct would be with substances that would basically merge to this anal pore and release. There's macro and micronuclei that lend to reproductive and somatic cell information with that. Contractile vacuoles that help to deal with osmoregulation, but really it's kind of this image with the mouth and anus or anal pore that I think are kind of key points on this. So as the foodstuffs, whatever it is, are being brought in through the cytostome, they wind up engulfed and in turn inside of a vacuole, all right, that merges with lysosomes, gets degraded, and then exported out, all right. So I think with that, I will stop. Any questions or concerns? To me, silence means no today. I think you're all good. All right. I'm hoping that all of this loads up onto YouTube. We will see. Have a good day, guys. I will see you all on Wednesday.